Good morning, everyone. A warm welcome to all the eminent personalities, honorable speaker of today's session, Professor K.V.Ramanamurthy sir, distinguished guests, fellow pharmacy colleagues, participants, dear students to this virtual webinar on biopharmaceutical and therapeutic consideration in dosage form design, organized by Sri Padmavathi School of Pharmacy, Department of Pharmaceutics, Tirupati. I, B.R. Srinivasmurthy, working as assistant professor in Department of Pharmaceutics, Padmavathi School of Pharmacy, take this opportunity to extend a hearty welcome to all the dignitaries to this event. Sri Padmavathi School of Pharmacy has a tradition of organizing national and international seminars every year, but due to this current COVID-19 pandemic situation, we couldn't organize this scientific session as an international seminar on campus. Hence, we are organizing a webinar to create a valuable platform for pharmacy professionals to gain the knowledge and to witness a useful talk by our distinguished speaker, Professor K.V. Ramanamurthy sir. First of all, let me start today's program with inviting dignitaries. First, let me take an opportunity to invite coordinator of today's program, head of the Pharmaceutics Department, Dr. V. Shanmugam sir to the session. Sir, please welcome sir. I extend my warm welcome to our organizing secretary for today's session, Head of Pharmaceutical Chemistry Department, Dr. C. Sridhar sir. Sir, please welcome sir.
Good morning, Sridhar.
I'm delighted to offer a heartfelt welcome to the convener of today's program, President of Indian Pharmaceutical Association, Tirupati branch, and Principal of Sri Padmavathi School of Pharmacy, Dr. D. Ranganaiklu sir. Sir, please welcome for today's session, sir.
Thank you. Very good morning. Very good morning to all. Thank you, Srinivas.
Now, it's my honor to invite Director of Sri Padmavathi Educational Institution and patron of today's program, Sri P. Pranit sir, for this webinar, sir. Sir, please welcome, sir.
Thank you, Mr. Srinivasmurthy.
Morning,
Thank
sir.
you. Thanks a
Morning.
lot. Good morning. Good morning, sir. Good morning.
I'm delighted to welcome Honorable Chairperson of Sri Padmavathi Educational Institution and Chief Patron of today's session, Srimati P. Sulochana Madam. Madam, because of her preoccupied schedule, Madam will join later. And Madam has conveyed her warm greetings and best wishes for today's program. Now, let us all wholeheartedly welcome, and I deem it a great honor to invite today's chief guest and honorable speaker of today's session, Professor K.V. Ramanamurthy sir, to this webinar.
Thank you.
Sir, we welcome you, sir. Thank you very much for accepting our invitation and sparing your most valuable and precious time for this session, sir. Welcome, sir.
Thank you.
Next, let us start today's program with invocation prayer song.
सरस्वती नमस्तुभ्यं वरदे कामरूपिणी विद्यारम्भं करिष्यामि सिद्धिर्भवतु मे सदा पद्मपत्रविशालाक्षी पद्मकेसरवर्णिनी नित्यं पद्मालयां देवी सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा.
Thank you, Charan. Now let me present glimpses of Sri Padmavathi School of Pharmacy very shortly. Sri Padmavathi School of Pharmacy was established in the year 1997 under the aegis of Sri Padmavathi Educational Institution, founded by late Dr. P. Mohan Rao sir, and currently headed by Chairperson Srimati P. Sulochana Madam Garu and Director Sri P. Pranit sir. And the courses offered in our college includes B Pharmacy, M Pharmacy, PharmD, PP, and R&D Center. And post-graduation branches available at our college includes pharmacology, pharmaceutics, pharmaceutical analysis, pharmacy practice, PharmD, and post-baccalaureate, and R&D center. Achievements in the last 3 years. Coming to the achievements in the last 3 years, we got a Campus Preparedness Award in the year 2015 and 8 Pratibha Awards and 1 University Gold Medal for the last 3 years. And research activities witnessed by more than 100 publications and 84 poster presentations in various national and international seminars and symposiums, and 8 PhD awardees from our institute. And one of the important exam, that is GPAT for our BPharmacy graduates. Every year, almost more than 10 students used to qualify in the GPAT examination. Looking at the year 2019, 8 students were qualified, and in 2018, 12 students, and in 2017, 11 students qualified for GPAT, and similarly got admissions into the one of the esteemed institute in the pharmaceutical field, that is NIPER. In 2018, 9 students joined NIPER. In 2017, 7 students, and 11 students in 2016. In addition to these academics, we also actively celebrate National Pharmaceutics celebrations and World Pharmacist Day, witnessed by the pictures, where we conduct various competitive competitions like elocutions, essay writing, and debate. We also conduct rallies on important days like World Diabetes Day and National World Pharmacist Day to bring the awareness regarding the public. And we also have a tradition of visiting industries every year. And currently, 10 research scholars are pursuing PhD at SPSP under various universities. In addition to this, we also have extracurricular activities like dance and music club, sports and games club, career guidance cell, placement cell, grievance cell, anti-ragging cell. As I already told you, we used to organize national and international seminar every year on campus. And we also celebrate the annual day every year. And Professor B. Suresh sir, PCI president, was also one of the guests for annual day celebrations. And social welfare activities, where we'll conduct health camps. And every year we invite the alumni for alumni meet. Thank you, sir. Next, I invite Director of Sri Padmavathi Educational Institution and patron of today's session, Sri P. Pranit sir, to address the session, sir. Sir, please.
Yes, thank you, Mr. Srinivas. Yeah, very good morning to one and all, and I welcome you all to the webinar on biopharmaceutical and therapeutic considerations in dosage form design that is being organized by Sri Padmavathi School of Pharmacy by the Pharmaceutics department. So let me congratulate the department and the whole team of Sri Padmavathi School of Pharmacy who have been organizing such kind of webinars continuously to update the knowledge and share the knowledge from the eminent personalities. So I would like to congratulate and thank the principal and the complete team of Sri Padmavathi School of Pharmacy for conducting such kind of webinars. And I would like to, first of all, a very good morning to dear students, faculty, team SPSP, and today's speaker, Honorable Speaker, Professor Ramanamurthy sir, Principal and Professor, Andhra University. Thank you, sir. First of all, let me thank you for accepting our invitation to be a part of us and to share your knowledge to all the students and all the research scholars and the faculty. And yes, these kind of seminars or webinars, whatever we call, because previously we were using the word seminars and now we are calling it webinars. So though we are not having any physical meets now, during this pandemic, I feel that many of us were feeling that we have been losing many things. But I feel personally, I feel that during this pandemic, we have got a very good time, because I think in the whole lifetime, I think we may not get this kind of free time. So I request all the participants who are here to utilize time to update your knowledge, to update yourselves, and to be mentally and physically fit, and to see that you can update yourself and see that a lot of changes you can get by thinking positively. So that is what I request, because you don't get again such kind of time again. And previously it was very difficult to conduct seminars because bringing speakers, eminent personalities, because it was very difficult for us to get that time to bring them down to our campus, have seminars and all. That was a bit difficult because they were a bit busy. So now with this pandemic, the technology, the use of technology is being more, so that 1 hour a day from their office, so that in sharing of the knowledge. So I think this pandemic thing, this pandemic, I think we should utilize properly.
Sir, kindly excuse us, sir. Respected Pranit, sir, we are unable to hear you, sir.
Sir, your voice is not audible, sir. Pranit sir. Might be it's a mute, might is in a mute, just unmute, sir.
Thank you, sir. Thank you very much for your valuable words and enlightening the importance of utilization of important session. Next, I request patron convener for today's program and principal of Sri Padmavathi School of Pharmacy, Dr. D. Ranganayakar, sir, to give the open remarks for today's session, sir. Sir, please.
Thank you, Srinivas, for inviting me as a coordinator on this part. Respected my professor and my guide, my mentor and philosopher throughout my academic career, that is Professor Ramanamurthy sir, and my management director, Sri Pranit sir, and my madam, P. Sulochana madam garu, correspondent, and organizing secretary, Dr. Sridhar, program coordinator, Mr. Dr. Shanmugam, all my team, and delegates from the registration part and entire the student scenario. It's a warm welcome and pleasant good morning to all on this webinar, free webinar program by Professor Ramanamurthy sir, Andhra University Principal and Department of the Pharmaceutical Sciences, specialization in pharmaceutics. And we have the organization Padmavathi School of Pharmacy, and we have the Padmavathi Education Institutions, all young birds of the pharmacy, both B Pharm, M Pharm, and pharmacy practice. Across the world, many registration has come. It's more than 500 registrations reached today from the different countries like Ethiopia and the many European countries also many registrations has come. Altogether, it's crossing more than 5 to 600 registrations so far. Really, it's remarkable. Looking at the title of webinar program by Professor Ramanamurthy sir, particularly on the topic of the biopharmaceuticals and therapeutic consultation in designing in designing form, dosage form. I feel it is a very important note for all the courses. Apart from this, just I welcome you all this webinar program. As you know that because many speakers has been highlighted this kind of importance of this webinar program, it's very opportunity for us to have a share of our feelings on the digital platform instead of become inert by looking at the COVID or the conditions on the present scenario. It is a kind of easy accessible platform where we can interact ourselves, we can update our knowledge, we can enrich the research activity on many titles. It's a very suitable title, especially designing on the farm, looking at the biopharmaceutical and therapeutic considerations. It's a really very wonderful program because whatever it may be the innovations in the part of any allied in the pharmaceutical side, our therapeutic activity is the main goal because to have the better therapeutic activity, these are the fundamental things to reach to consider before forming a design because it's very much interlinked with a posology and the consideration of biopharmaceutics and the dosage farming. Otherwise, all our efforts are going to be in vain, the waste of time, and it is going to progress the disease. And simultaneously, we cannot reach the progress in the innovation of any research-oriented work. This is quite useful for the right from beginning of UG to PG and PhD program. That's what I selected this program with the request of Professor Ramanamurthy sir. As I said earlier, he is my professor during my pharma and he is my guide during my PhD program. He is my philosopher entered my career right from beginning of this institute. As Srinivas has said, is 97, 98 established right from beginning of this institute development. He is also one of the part and parts of this advisor to develop this institute. This is today I'm here. This coincidence is grace and well wish for this kind of progress. I'm very much thankful for Professor Ramanamurthy sir to accept of this program and coming over on the right time. Because it is right time is just I would like to quote, I take the opportunity on this occasion. During my program of BPharm, I mean MPharm at the Andhra University level, not me, a majority people have been considered is marked within a kind of the discipline and punctuality and other things. People used to consider him as a bond. In sense, he is very particular on the time sense and he is very particular on the commitment of work and he is very compatible and coordination with the student problems and salvation of problems and updation of the knowledge. That is the reason he's very active, enthusiastic, and we are very much being fascinated looking at his style of work and now teaching age that he has reached the hearts of students as a working encyclopedia in the University of Pharmaceutical Sciences, Andhra University, and he has very much remarked for the time sense and punctuality. He used to conduct a bond like that is remarkable. No credentiality. What I noticed during my course of MPharm and PhD work. This is my association with Professor Ramanamurthy sir for more than 23 years. In this, always I used to saw such kind of his courtesy and considerations student community. Whenever we ask anything, he don't say no because I know what I'm quoting here. You might be all knowing that the SAR is going to reach the superannuation on 30th. So Sar is so busy with many official busy schedules and his own traditional formalities. When I'm asking, because my eagerness is that I don't want to lose my professor voice before in sense, losing sense. I want to make use of his voice and updation of knowledge before his retirement. Of course, after retirement also the same continuity will be there. But even then, I had a sentiment. I'd offer his services to give a kind of lecture during this COVID program but he readily accepted keeping all his preoccupation schedules aside. He has given such kind of assurance. He has been here at the right moment, at the right time to educate and to update all the students. It is not only confined for the SPSP. SPSP organizing this for the benefit of all the pharmaceutical sciences graduates, postgraduate, and PharmD fellows in the regard of biopharmaceuticals and therapeutic considerations in designing pharma. So with this, I convey once again thanks to Professor Ramanamurthy sir for this readily acceptancy, and I wish you all the best for the program, especially delegates who have been eagerly waiting for the program. Definitely, I hope this program is going to be clear that all your clouds and clear the almost your critical gap in between the research and academic area, and it will enrich that your knowledge regarding the dosage farming. Thank you very much, Mr. Srinivas.
Thank you very much, sir, for present the opening remarks of today's program. Good morning to all of you, to the respected dignitaries. Myself, P. Keethasika, Assistant Professor of Department of Pharmaceutical Analysis, immensely pleased to present the profile of Professor K.V.Ramanamurthy sir, Principal and Professor, University of Pharmaceutical, Andhra University, Visakhapatnam, to the virtual audience. An iconic personality in the pharmacy profession, a best teacher, researcher, an eminent and cognizant speaker, and who always encourages all the scholars, faculty, and student fraternity by radiating the knowledge and creating the awareness about the novel departments in the field of pharmacy. So now let me introduce the sir to this today's session. Professor K.V.Ramanamurthy sir completed his graduation, post-graduation, and PhD from the Andhra University, Visakhapatnam. Sir served as lecturer, associate professor, and senior professor and principal of Andhra University, and also sir held many professional positions like chairman of BOS of Andhra University, member of AP Pollution Control Board, academic coordinator, and member of NAC steering committee, as well as placement and training officer in the AU College of Pharmaceutical Sciences. Sir has guided more than 65 PhD students and 120 MPharm students, and Sir has contributed enormous to the research field by publishing more than 180 research papers at the national and international level journals with high impact factor. Sir has got 4 patents in the department in the field of pharmaceutics. So with the Sir's contributions, Sir was awarded with many awards like Best Research Award by APTI in the year 2019, State Teacher Award by the Government of AP in the year 2015, Dr. Sarvepalli Radhakrishnan Best Academician of the Year by Andhra University in the year 2010, and Sir was a recipient of APTI Teacher Award in 2009 by Association of APTI, and Sir was awarded with AP Scientist Award by Andhra Pradesh State Council of Science and Technology and the Best Research Award by Andhra University. And also Sir got this international study and research grant for conducting the research in a foreign country for a period of 2 months with a grant of rupees 1 lakh by Andhra University 2004. And also Sir was awarded with UGC PG Fellowship as well as UGC JRF. So with this brief profile, I would like to invite Professor K.V.Ramanamurthy sir to address the virtual gathering. Really, once again, thank you, sir, for accepting our invitation and being with us today.
Yeah, very good morning to all of you. In fact, I'm very much indebted to the Padmavathi School of Pharmacy for the nice words that are spoken about by the principal, my student Ranganayaklu, and Ms. Preeti, assistant professor, and director, Mr. Prof. and other faculty. I can now start sharing my PowerPoint, right? I think you are able to see my PowerPoint, please.
Yes, sir. It is visible, sir. Yes, sir.
Yeah.
Visible, sir.
Yeah. Today's topic, what I have selected for the talk is biopharmaceutical and therapeutic considerations in the design of dosage forms. And according to my information I received, majority of the students of B.Pharm, M.Pharm, PharmD, and PhD scholars. And biopharmaceutics is a subject many of these youngsters used to dislike, I don't say hate, but they used to dislike this subject for one important reason, that is the mathematics involved, that is the calculations of various kinetic parameters. But however, it plays a very important role in deciding a type of a dosage form that is to be administered to a patient for exerting the required pharmacological effect of the drug. So if you just look at the introduction, in the basic parts of my slides, I will try to refresh your mind with the basics of biopharmaceutics, what you might have studied earlier. Don't curse me, but it is very important to refresh the knowledge always to learn the advanced knowledge. So if you just look at this, throughout its history, pharmacy profession is concerned primarily with the manner in which drugs produce their pharmacological effects. That means the basic research always concentrates how a drug is going to produce the required therapeutic effect in the name of pharmacological action. Once a required pharmacological action is produced, the duty of the pharmacist is to convert that into a suitable dosage form through which we are going to administer the drugs to the patients. So, lot of emphasis has been laid on the establishment of the physicochemical properties of the drug and their relationship with the pharmacological responses. So, these properties are normally termed as biopharmaceutical properties. The biopharmaceutical properties of the drug substances include majorly the solubility, partition coefficients, diffusivity of the drug, degree of ionization of the drug, polymorphic nature of the drug, etc. So the biopharmaceutical properties of these drugs must be very much important, and they contribute an important factor in facilitating the absorption and distribution process and how they are going to reach to the active sites. If you look at the definition, which is the basic and primitive definition given by John G. Wagner, it is the study of influence of formulation on the therapeutic activity of a drug product. And it is the study of relationship between some of the physicochemical properties of the drug and its dosage forms and the biological effects observed following the administration of the drug in various dosage forms. So if you look at this definition a little critical, you can see that it is going to create a relationship between the physicochemical properties of the drug and how these physicochemical properties of the drug are going to influence the design of dosage forms and the respective biological effects that are produced after the administration of the dosage form is very, very important. And if you try to alter any of the drug delivery system, it is going to alter the rate and amount of the drug that is going to be delivered to the desired place in the body. So what are these alterations we can expect? A slight change in the chemical nature of the drug, the particle size and surface area of the drug, the type of the dosage form, and other manufacturing processes, what are being used in their manufacture. If you look at the process when the drugs are administered, majority of the drugs, almost 85% of the drugs are preferred to be administered through oral route. That's why my emphasis on this dosage form design is majorly concentrated on oral route compared to the other routes of administration, because the design is very, very important and critical. We may think design of oral dosage forms is very easy compared to parenteral or other types of dosage forms like transdermal or nasal. But in my opinion, as a pharmaceutical researcher, I feel designing a very good oral dosage form, circumventing all the problems that are going to come during the transfer of drug from the site of administration to the site of application, one has to answer so many queries. So if you just look at this, whenever a drug is given through oral route, it may be subjected to the various pH conditions. What is the volume of the fluid that is contained in the stomach, residence time of the drug in the stomach, the effect of food, and when you consider the type of the dosage form, how the dosage form, like a solid dosage form, is going to deaggregate or disintegrate, followed by the dissolution, that means the rate and extent of the drug release from the dosage form. And during this process, even though the drug is released from the dosage form, there is a chance of formation of complexes with the mucous membranes or mucus that is present in the stomach. And there are some amines that are going to be there which may complex these drugs. And when once it is transferred from the stomach into the small intestine, there is a chance of precipitation of bases due to the slightly alkaline conditions, because majority of these drugs are weakly basic or weakly acidic drugs. Hence, when they go into the basic side of the intestine, there is a chance of precipitation, and we have the bile salts that are getting secreted from the bile. So there is a chance of solubilization effect by the formation of the surfactant moieties due to the interaction of the bile, cholesterol, and others. There may be a chance of improvement in the solubility. There is a chance of complexation, maybe a positive complexation or a negative complexation. And all these are the various factors. Besides that, the entire GIT is connected through portal vein. Majority of the people believe that the first pass effect that is observed due to the is because of the transfer of the drug from the stomach to the liver through portal vein connected to the stomach. But portal vein is completely innervated right from stomach to almost near to the rectum part of the GIT. So there is every chance that the drug which is getting absorbed all through the GIT may be first landing into the liver, where there may be a chance of metabolism, and only the drug that is escaping this metabolism may go into the blood circulation. So one need to consider all these parameters before thinking of delivering the drug through a particular dosage form. So when a drug is to be absorbed into the systemic circulation, it requires a passage into in the form of a molecular form. That means almost in the solution state, except the nanoparticles of very small size entities. All other drugs must be first converted into a molecular form in a solution state, and it should be able to cross the biological membrane, which is going to act as a barrier. So the first prerequisite is release of the drug from the dosage form. So the drug particles must dissolve, and if they possess the desirable properties what I have mentioned earlier, we can expect a high concentration of the drug at the region of the absorption, where the drug is transferred from higher concentration to a lower concentration, that is nothing but blood into the general circulation. So when we consider the effects of molecular properties on solubility and permeability, the physical form of the drug is very, very important. And there may be a chance of melting point influence also sometimes. That means physical form is going to be influenced by the melting point, and it may be directly going towards the solubility, or through melting point it may go into the solubility. And once the drug is soluble, there is a chance of charge development on the molecule due to the weakly acidic or weakly basic nature of the drug. And whenever there is a charge development, it may be ionized, and there is a chance of formation of hydrogen bonding. And depending upon the solubility, the molecular size may be a larger size or a smaller size, and all these are going to influence the permeability of the drugs through the biological membrane. And if it is molecular size is bigger, what is the shape of that molecular size, and how the charge is distributed on this particular. And finally, we think about the absorption properties of the drug. If you look at the biological membrane structure, it is a bilayer structure consisting of the phospholipids, out of which the hydrophobic heads are pointed towards the inside and hydrophobic heads are pointed towards the outside. And we have the sugar side chains that are present on the surface of these molecules, and the integral proteins are going to form a channel-like structures, and it is innervated with the water-filled pores with a very small diameter of 4 to 5 or 4 to 10 angstroms. And all these are going to play a very important role in the transportation of the drugs from the site of absorption, site of administration, to the systemic circulation, that is site of absorption. So one need to take into account about all these parameters, because this hydrophobic layer may be having fatty acid tail, phospholipids, etc., which is highly lipophilic in nature, and the superficial layer is highly hydrophilic in nature. So depending upon the type of the drug that is being administered, these are getting activated and aid in the transfer of the drug from the administration site to the absorption site. So how the drugs are going to be absorbed into the systemic circulation? We all know these basic things like passive diffusion. So a molecule move from a higher concentration to a lower concentration is called as the passive diffusion, and it is pictorially represented here. That means this is a high concentration that is getting transferred across this biological membrane to the lower concentration site. Sometimes we have the protein molecules that are present, and the drugs may be transferred across these protein molecules because of this lipophilic nature also, it may happen. So the movement is caused by kinetic energy of the drug molecule, because there will be always in a continuous motion, the chance of development of a kinetic energy is there, and hence they may be transferred by the kinetic energy of the drug molecules. And it is a concentration driven process depending on the first order kinetics. And the parameters that influence this passive diffusion is membrane permeability, which is dependent on the lipophilicity of the drug molecules. That means there should be an optimum lipophilicity and optimum hydrophilicity for a drug to be transferred across the biological membrane, because the superficial layer is hydrophilic in nature and internal layer is lipophilic in nature. When the drug is crossing this, first it should have a hydrophilic interaction with the superficial layer, and when it is penetrating into the inner core of that particular biological membrane, the lipophilic activity is going to influence its transfer. That's why we need to always have balance between the hydrophilic and lipophilic nature of the drugs. The second important mechanism that involved in the transport of drugs is the carrier-mediated transport, and actually it is subdivided into two major important transport mechanisms. One is called as passive facilitated diffusion. The second one is called as the active transport. But in majority of the cases, we call it as active transport colloquially. And some of the drugs are absorbed by the action of a transporter proteins, which are going to act like a carrier, or some of the enzymes that are present in the GIT, they may act like the transporters for these special class of drugs. And for the requirement of this active transport, there should be a structural similarity between the drug and the substance or substrate what is going to transfer this particular moiety. And it is going to occur against a concentration gradient, unlike the passive diffusion, because here we are involving the energy in the form of ATP for the transportation of the drugs. And my dear young fellows, I'm going to give a small clarification to all my B.Pharm or even M.Pharm students. Whenever we ask a question in the Viva, what is active transport? Involves ATP. So I just say a small joke here. I give you 10 liters of petrol can without a vehicle. Can you travel? You cannot, because you have energy in your hand like petrol, but without pouring this petrol into a vehicle like a two-wheeler or a four-wheeler, it is practically impossible to cause the motion. So though ATP is involved in the transportation of the drugs by using the active transport, there should be a carrier which should aid the transport mechanism, and hence please ignore the first 2 pages of the Brahmankar book, what you are accustomed for reading, and flip the remaining pages beyond the first 2 pages of the absorption mechanisms. There, Brahmankar has beautifully explained what is the role of a carrier in the transport mechanism. And because the number of carriers are limited in the body of different parts, it is called as a capacity limited, and it follows a non-linear kinetics, because before the concentration of the drug is more than the number of carriers, the mechanism or kinetics follow first order. Once more drug molecules and less number of carriers are present, there are no carriers. It is just like when you want to cross a river using a ferry, unless the ferry is, if the number of passengers are less than the capacity of the ferry, all the passengers can easily cross. But when once the ferry is reaching to its limit of carriage, and if there are 50 passengers and the capacity of the ferry is only 25, the ferry can take only the first 25 who came, and the remaining 25 have to wait. So in the case of ferry, we can wait. But in the case of active transport, the drug cannot wait, because in the body, it is not a static process. It is a dynamic process, because the drug always moves from one part of the body to the other part of the body in the absorption site, and it's slowly being pushed from the stomach to small intestine to the large intestine very continuously. And once the drug misses this particular carrier site, like which we call it as absorption window, it is possible that the drug may not be absorbed completely. Hence, the novel drug delivery system of gastro-retentive drug delivery was designed, where we create a depot in the stomach and the drug is slowly allowed to release, wherein it reaches to the small intestine, which is the major seat for these carriers. And because the number of drug molecules that are reaching to this particular site are less than the carriers that are present, and they can be easily absorbed. So vitamins and some of the drugs like propanol hydrochloride, metformin hydrochloride, etc. These are the drugs which can be delivered by using these types of specialized methods, wherein we can titrate the drug release in such a way such that the drug concentration is always less than the number of carriers that are present for its absorption. And we have one more absorption mechanism called as convective absorption. So the lipid layer, as I mentioned already, is having a water-filled pores with a radius of approximately 4 angstroms, and some books may say it is 4 angstroms to 10 angstroms, whatever the case, the molecular size of the drug, if it is less than this particular size of 4 angstroms, they can readily pass through water-filled pores. But the second criteria required for this is the drug should be always hydrophilic, highly hydrophilic, because the pores are filled with water, and unless the drug is highly hydrophilic, it cannot dissolve and pass through this particular pore. And this is called as the convective absorption. And there is one more parameter that is called as sieving coefficient. So sieving coefficient is a parameter that is used to assess the capability of the drug to cross this biological membrane, where the concentration at the receiving end divided by the concentration at the donor side. That means CR by CD, we can calculate the sieving coefficient, and it is also indirectly proportional to the relative sizes of the pores and relative sizes of the molecules. So these are the parameters that are going to influence the convective absorption. So ion pair absorption, we have highly ionized compounds like quaternary ammonium compounds, which are ionized in almost all pH ranges, but they are still used as drugs. So the mechanism of these types of ions that are going to be absorbed is by forming a complex which is a neutral in nature, and it is going to form a complex, a neutral complex with the endogenous ions of the GIT, and they create both the required lipophilicity and as well as aqueous solubility. And once this complex is formed, they are being transported by passive diffusion, and after reaching to the site of absorption, site of action, they'll again uncomplexed with this particular ion, and they again be available as a free drug. Then coming to the physicochemical factors that are going to affect the drug absorption are the pH partition hypothesis of a drug absorption. So if you want to look at the drug action in the basic sense, the first criteria for a drug to be effective is its solubility, because the drug must be available in the form of a solution, because to create a molecular size. Once the drug is in a soluble state, the second important criteria is its unionized nature, because majority of the drugs, they will be absorbed only in the unionized state. So if a weakly acidic drug is taken into a weakly basic environment, certainly there is going to be an acid-base interaction between this weak acid and weak base, whereby they are going to be in the ionized state. But a weakly acidic drug is kept in the environment of a highly acidic environment. The weakly acidic drug cannot be ionized. The one which you have studied in your ionic equilibria chapter of the physical pharmacy in your basic class of first year or second year. So that is going to be a very important parameter. So based on this, the second important parameter is partitioning effect. As I mentioned about the biological membrane, it consists of two important layers. One is the hydrophilic layer, external, and lipophilic layer, internal. So the drug must be able to have a partitioning effect. That means like a benzoic acid partition between benzene and water, we have to have a partitioning effect of this drug between the hydrophilic environment of the superficial layer, where it will be attached to that, and once it is attached, it slowly passes down to the lipophilic layer. Again, there is going to be a partitioning effect, and it comes out of this layer, other side, again, which is hydrophilic in nature. So for causing this partitioning effect, the partition coefficient of the drug is going to be very, very important. So clubbing these two parameters, we call it as pH partition hypothesis on drug absorption. So the gastrointestinal and other membranes acts like lipid barriers, and the unionized form of the acidic or basic drug is preferentially absorbed, and most drugs are absorbed by passive diffusion, and the more the lipophilicity of the drug, the faster is its absorption, and weakly acidic and neutral drugs are absorbed from the stomach, but basic drugs are absorbed from the other parts of the body, that is nothing but small intestine or the large intestine. So we can have a good correlation between the ionization and pH at the absorption site based on the Henderson-Hasselbalch equation, what you have studied in your basic classes, which states that pH is equal to pKa plus log salt by acid, which is slightly modified for biopharmaceutical point of view, which states that pH is equal to pKa plus log salt log ionized by unionized or unionized by ionized for the acid and base respectively. So the acid dissociation constant of a drug is going to be very, very important, and we have the acid dissociation constant of majority of the drugs. So it is nothing but the negative logarithm of dissociation constant, and by looking at the pKi value of a drug, we can have an easy assessment of the place from which the drug is going to be absorbed from the GIT. For example, the equation, let us take it for the weakly acidic drug, pH is equal to pKi plus logs ionized by unionized, and if the pKi of the drug is 8 and pH of the stomach is approximately, let us take it as 2 is equal to 8 plus log ionized by unionized. So if you bring this 2 and take this other side of the RHS and LHS exchanges, the log ionized by unionized is always a positive sign. So it indicates that the pH is not going to influence the absorption of the drug, and we assume that all the drugs which are having a pKi value of more than 8 can be absorbed all through the GIT, because there is no ionization of the drug at all in any given pH region. So in the similar way, we can predict for both acidic as well as basic drugs how they are going to be absorbed based on the pH conditions of the GIT, which is an important parameter one should think before going for a design of a dosage form. For a weakly acidic drug having a low pKi value, we can imagine that it may be ionized at all pH conditions, and hence its absorption is going to be a big issue, and we need to design a suitable dosage form to overcome this ionization parameter. And these are some of the pKi values that are being given for these drugs. And second one is the lipid solubility. As I mentioned, the lipid solubility is going to be a very important parameter, and we can calculate the partition coefficient, and normally the partition coefficient value is calculated for biological purposes between the two systems, namely N-octanol and water or chloroform and water. Preferably the first one is more preferred compared to chloroform and water. But we consider this logarithmic value of this partition coefficient, and if this log p value is more than 1, it is highly lipophilic in nature, and if the log p value is 1, it is both 50% lipophilic, 50% hydrophilic. And if it is negative side, it is more hydrophilic rather than the lipophilic in nature. So it is going to influence the transport and the absorption process of the drugs, and we can use a structure activity relationship by using various approaches. So for a practical viewpoint of drug, as I mentioned already, there should be a proper balance between the hydrophilicity and lipophilicity of the drugs. Then how we can say that the drug is absorbed into the systemic circulation? There should be a quantitative measure rather than a qualitative measure, and we have the term called as bioavailability. And we all know the definition. It is defined as the rate and extent to which an active ingredient or an active moiety is absorbed from a drug product and becomes available at the site of action, and which may be available for absorption due to the reasons like chemical degradation. It may not be available if it is degraded chemically in the stomach itself, for example, penicillins. Physical inactivation because of binding and complexation with substances in the intestinal tract. Tetracycle is one of the best example. Incomplete dissolution of dosage forms. We all know majority of the drugs are almost partially or practically insoluble, and hence dissolution is going to be a very important rate limiting step. Microbial biotransformation. Majority of the drugs may be degraded due to the microbes that are present in the, especially in the large intestine, and there may be a chance of drug degradation. Insufficient contact time in the gastrointestinal tract. As I mentioned, the stomach, sometimes it is resided in the stomach only for 1 to 2 hours, and it may not be absorbed completely because the absorption site itself is stomach for that particular drug. Poor solubility, poor permeability, metabolism within the gut wall. We all know the first pass effect where the drug is affected by the liver, but some drugs like oxytocin, the moment it is given into the stomach, it gets deactivated by the stomach enzymes as well as the acidic environment of the stomach. That's why the half-life of oxytocin is not more than 2 to 4 minutes. So sometimes even before reaching to the gut wall, the drugs are getting degraded, or in the gut wall itself by various enzymatic actions and other degradation pathways, the drug may be degraded. So all these are the various obstacles that one may have during the estimation of bioavailability, and we need to identify the right cause. And this is how we are going to estimate the bioavailability, that is fraction of the orally administered dose that is ultimately reaching to the blood circulation, and we can calculate by using FA, FG, and FB. FA is fraction of intact drug that is absorbed. FG is fraction of intact drug that escaped in irreversible elimination of the drug, passes subsequently from the GIT across the gut wall, and a fraction of the intact drug that traverses liver into the systemic circulation. That means it is escaping the liver metabolism. So there are three different factors that contribute for the total fraction of the drug that is reaching to the systemic circulation. So we have some misconceptions regarding the drug absorption. One is intestinal absorption, permeability, fraction of drug absorbed, and bioavailability are equivalent properties and can be used interchangeably. Because once we assume that it is getting absorbed, we assume that it is reaching to the systemic circulation. It is permeating across the biological membrane, we assume that it is reaching to the systemic circulation. Fraction of drug absorbed or bioavailability, we assume that it is absorbed. Absorption, permeability are discrete fundamental properties of drug molecules and can be predicted solely from its chemical structure. So this is also another misconception. But in reality, when we face the on-site problems, the solubility, permeability, formulation factors, and physiological variables such as permeability differences, pH, luminal and mucosomal enzymology, intestinal motility, all are going to play very important role in deciding the bioavailability, permeability, absorption, etc. We have a very popular rule which is called as Lipinski's rule of 5, and according to this rule, poor intestinal absorption is associated with and attributed to the molecules possessing any two of the following properties. So whenever the molecular weight is greater than 500 to 750 Daltons, or number of hydrogen bond donors greater than 5, number of hydrogen bond acceptors greater than 10, and the calculated log P is greater than 5. That means the drug is highly lipophilic in nature. So based on this Lipinski's rule of 5, by looking at the chemical structure itself, one can easily assess whether it is a highly hydrophilic or highly hydrophobic drug, and if any of the two characteristics that are there for this particular drug, it gives a problem for its intestinal absorption, and accordingly proper care need to be taken during the dosage form design. And another important property what we encounter is polar surface area of the drug. So there is a relationship between the molecular surface properties and biological performance of a molecule, and PSA of a drug molecule has a strong correlation of predicting the drug transport from human intestine and across the drug membrane, because as the PSA is increasing, the forces like van der Waals surface area for the polar atoms, oxygen, nitrogen, and attached hydrogen atoms will increase. That means the number of H-bond donors or H-bond acceptors will increase, and it deviates the rule of Lipinski of 5. And it is a major determinant to for oral absorption and brain penetration of drugs. Because for oral absorption, hydrophilic is more preferred. For brain penetration, highly lipophilic drugs are preferred. So how we can measure the permeability of a drug? So in the recent times, there are different cell layer models were developed, which are called as cell monolayer models. These models consist of cells grown on permeable inserts. So transport of compounds across these cell monolayers can be used for quantitative estimation of the permeability of the new chemical entities in a very quick manner. Of course, sometimes when we go into the real in vivo experimentation, there are chances of deviation, but still we can presume and assess the permeability of majority of the drugs which are under the development stage. So the popular cell lines that are normally used for assessing the permeability of the drugs are the COCA2 cells, and they are derived from human colon adenocarcinoma cells. So the monolayer has a very specific character called as ion conductance, and it possesses the trans-epithelial electrical resistance, which indicates the formation of tight junctions that are there in the normal membrane, and it can be used for the assessment of cellular transport for a chemical entity very effectively. The second type of cell lines that are being used are Madin-Darby canine kidney cells, MDCK, And though we are using coca-2 cells to a great extent, these are gaining popularity in recent times because the shorter culture time, 4 to 7 days for these MDCK cell lines, whereas 21 to 30 days for coca-2 cell lines. And hence, they are going to be used more frequently nowadays. And some researchers have carried out a study for calculation of the permeability coefficients by comparing MDCK and coca-2 cells, and it was observed that 55 compounds with known human activity have given similar results, and there is an excellent correlation between MDCK cell lines and coca-2 cell lines. We have one more important system which can characterize the drugs basically without going into the wet lab by using a system called as biopharmaceutical classification system. So many of my friends might be knowing all this, but I'm still repeating. Please bear with this because some basics are always important for repetition every time. So we can allow the prediction of the in vivo pharmacokinetics of a oral immediate release drug product by classifying the compounds into 4 classes based on 3 important parameters. You may be knowing the third important parameter, which was recently added by FDA to the first 2 important parameters, namely solubility and permeability, which was originally proposed by Emidon. But later, US FDA has introduced one more important parameter called as the dissolution properties of the dosage form. So it may be due to the low bioavailability, may be due to poor solubility and low permeability, and it is the basis for the classification of the drugs. So I'm not going to name the classes, class 1, class 2, class 3. It may come in the next slide for the just information sake. But what do you mean by high solubility criteria? Very, very important. Many of the young friends may be having a misnomer that the solubility means it is highly soluble, but there is a quantitative expression in the BCS classification for the high solubility criteria where we have to take this highest strength of the dose and it should be soluble in 250 ml or less of aqueous medium over the pH range of 1 to 7.5 at 37 degrees Celsius. So you can see the conditions that are laid down. One is the quantity of water or medium having different pH ranges starting with 1 to 7.5 because the oral pH starts from 1.2 to approximately 7.5 at different regions. So the drug must have the solubility in the entire pH range. Preferably we use a biological buffers suitable for the normal solubility determination. And why it is 250 ml? The volume estimate of 250 ml is derived from the typical volume of water that is consumed during the oral administration of a dosage form. As per American standards, it is approximately 8 ounces, and 8 ounces in our calculation is equivalent to 240 ml. What is the high permeability criteria? A drug substance is considered highly permeable when the extent of absorption in humans is 90% or more of an administered dose based on mass balance or compared with an intravenous reference dose. So, unless it shows a 90% bioavailability, the drug may not be considered as highly permeable. And the third parameter that is introduced recently to this classification system is the high dissolution criteria. So, according to this, a drug that should be dissolving rapidly by not less than 85% of the labeled amount of the drug within 30 minutes. That means the dissolution must be able to release. The 85% of the drug must be released from the dosage form within 30 minutes of the dissolution testing. So this is the classification that is divided into 4, and you can see the new chemical entities that are coming into the market. Approximately 5% are only belonging to the BCS class 1, and BCS class 2 they are about 70%, and class 4 only 20%, and BCS class 3 there are only 5%. That means highly soluble and low permeable. So this is an indication that what a formulation scientist is going to face a challenge for the design of dosage forms for overcoming the solubility criteria of the drugs, because majority of the drugs are poorly soluble, and hence the primary challenge a formulation scientist is going to face is how to increase the solubility of this particular drug. And one more classification was introduced approximately in 2008 by Bennett, and it is called as Biopharmaceutical Drug Disposition and Classification System. The objective of this particular classification system is, besides the BCS classification system, which defines only the solubility and permeability, the rate of metabolism is also going to be very, very important, because once a drug reaches into the systemic circulation, the rate of metabolism should be commensurate with the rate of input of the drug into the body. So some drugs which are going to have the extensive metabolism, they may be required for number of frequent doses. A drug that is having a low metabolism or poor metabolism may be requiring the dosage regimen or frequency of dosage administration at a more long intervals. So based on this criteria, BCS drugs are divided into, again, class 1, class 2, class 3, and they assumed that class 3 drugs are having poor metabolism, whereas class 1 drugs are having extensive metabolism. So it was proposed by Bennett in 2008. He's presented this paper in AAPS at America. So these are the various class 1 drugs. So the rate limiting steps for this class 1 drug absorption are gastric emptying, which is going to be the rate limiting step, and interactions may delay the gastric emptying, and they may delay the drug absorption for the drugs having the absorption in the small intestine, and they have given the different examples of class 1. So class 2 drugs, always dissolution rate is the limiting step for the absorption of the drug, and it will be griseofulvin, for example, it is having a very high dose and incomplete, whereas digoxin, it is having a very low dose, and hence dissolution, though it is poorly soluble, absorption is not completely affected and it is almost complete. So dose is also going to be very important for a poorly soluble drug. If it is a very low dose drug, it may not be a rate limiting step and it may be absorbed completely. So we have to consider the constant volume of the gastrointestinal fluid, what are the other co-administered substances like meal and other materials that are given along with this drug, what is the biliary solubilation effect of this drug, and what is the increased rate of gastrointestinal calorific density, etc., must be considered for these class 2 drugs. Hence, class 3 drugs. For drugs possessing high water solubility, the permeation is going to be a rate limiting step, and sometimes there is a region of absorption, what I have mentioned already, and we have to have some interactions also, and we can see sharp decrease in absorption when it is co-administered with meal that is independent of fat content. So all these parameters are to be taken into account for class 3 drugs. Class 4 drugs are much more problematic, and we have drugs like furosemide, paclitaxel, norfloxacin, etc., and we can increase the plasma concentrations for the drugs by improving their solubility as well as their permeability. And it will be useful for the drugs which are not having any narrow therapeutic index. But if the drugs are having a class 4 drug with a narrow therapeutic index, the formulation scientists will face much more critical challenges. So these are the rate limiting factors for the different classes of drugs. If you look at that, for type 1, the rate of gastric emptying, intestinal transit time, and for type 3, the transit across the columnar cells, and dissolution rate is the rate limiting step for type 2, and type 4, the parameters relating to type 2 and type 3 are going to be the problems of the type 4. Role of transporters in drug absorption is also another important parameter which we have to take into account in the formulation design. So we have the intestinal membrane transporters which may be affecting the rate of oral absorption or drug efflux into the intestinal lumen. So these are the different transporters that are going to influence the rate of oral absorption, and these are the drug efflux transporters that are going to influence the drug absorption, like PGP, MRP2, BCRP, MRP3, etc. And we have different intestinal enzymes which are going to convert the drug into very different types of the metabolites by using conjugation reactions, and that also to be considered, like CYP3, AO4, etc. So we have to consider the parameters like inhibition of membrane transporters when they are interfering with the absorption or metabolizing enzymes. So we have to take necessary steps for their inhibition, and we have to modulate the expression of membrane transporters or metabolizing enzymes. So these are the two approaches normally to be adopted when these enzymes are going to, or transporters are going to influence the drug absorption. Sometimes a new approach, we call it as a new always, but it is one of the oldest approaches that is available for altering the drug absorption, which is called as the pro-drug approach. So drugs that are having very low lipophilicity due to presence of polar functional groups, where there is no adequate balance between the hydrophilicity and lipophilicity, we can change that, and we can change them into a lipophilic prodrug systems or self-emulsifying prodrug systems to change their bioavailability by decreasing their solubility, which may be limiting the dissolution in the gastrointestinal tract, or increasing the dissolution rate, or improving the permeability across the biological membranes. And sometimes we can overcome this first pass metabolism, because whenever we are giving the self-emulsifying drug delivery systems, there is a formation of emulsions by the lipid proteins and other bile salts that are forming a crude emulsion, and with the extraneous surfactant that is added to these lipid drug delivery systems forms a fine emulsion in which the drugs are entrapped. And because they are highly lipophilic in nature, they will be directly taken up by the lymphatic systems, overcoming the first pass metabolism through the portal vein, and they will be directly landing into the blood circulation, whereby we can avoid the first pass metabolism. So by using the micellar solubilization approach or increasing the dissolution rate by these MET sets, etc., we can improve their absorption characteristics or prodrug approaches. So this is one of the best prodrug approach that was recently tried, which is famiciclovir, which is a dihydroxydiestate prodrug of penciclovir. So when we take the penciclovir, the bioavailability is approximately 4%, and when it is converted into a prodrug famiciclovir, when the dose is given, the bioavailability was significantly improved to 75%. That means after reaching into the blood, that is getting converted into penciclovir. So these are the various drugs that are coming under the different classes with a very poor oral bioavailability. So you can see almost all are less than 25 to 30%. So what are these biopharmaceutical properties, what I mentioned in the beginning of my talk, are going to influence the dosage form design? So they are physical, which are consisting of physical state, solubility, crystal form, particle size distribution, surface area, and physical stability. So chemical properties are salt forms, molecular weight, pKa, chemical stability, compatibility with excipients. And biological properties, mechanism of absorption, half-life, site of absorption, first pass metabolism, efflux metabolism, enterohepatic circulation, drug dosing requirements. So I'll just tell you a small interesting story how a formulation scientist fails in his research. Almost, I can say about 35 years back, one of the persons who graduated with MPharm started an industry and producing some drugs. It is a small scale venture. So he got a product of suspension to be manufactured on loan license from a big player, a pharmaceutical company, which is a pediatric formulation. And this person started making it on the loan license basis and used to give it back to the manufacturer. He made it like that for about 3 to 4 years. And he was lured by the profits that big pharmaceutical company earning on this product. He felt that I know the entire formula of that particular company, manufacturing process of that particular company. So why can't I float my own product for the same type of dosage form with another brand name of my company? At least I can take 5 to 10% of the business of that particular big player. So because the raw materials are also available for doing the formulation research, he developed his own product and pushed that into the market by procuring the drug. Unfortunately, within 1 to 2 months itself, the entire lot, almost 90% of the total batch went into the market, was returned back on the complaint of sediment formation of the suspension, which is a hard cake. He got confused. I followed the same procedure, but the batch is failing. So he repeated it once again for 3-4 times, and all the 4 batches were returned back to the manufacturer with the complaint of hard cake formation. And during the course of the manufacture, in those days itself, he lost about 10 to 15 lakhs on this product, which is not a small money nowadays. In those days, I can say. So he started investigating the cause for the failure. He is getting very good results for the big player, for the same formulation with the same contents. But for him, it is always failing. Then, using his friends in that big company, he started reasons. Unfortunately, he is procuring the bulk drug also from the same manufacturer who is a bulk drug manufacturer for the drug. And on investigation, it was found that the big player is making 2 different types of bulk drugs. In the last stage of the manufacture of this bulk drug, the crystallization process is getting altered for the outside commercial bulk drug and the inside requirements. So because of this change in the process of crystallization, whatever bulk drug that is sold to the outsiders of the company, it is going to have a different crystal structure and polymorphic nature, where it forms a big crystals and it leads to the caking formation. Whereas in this for this particular company product, they are taking special care in making a crystal where there is no chance of formation of a cake. Thereby, their product is very popular in the market compared to their competitors. So, though the drug is the same, the rate of crystallization, the nature of the polymorphic form, the nature of the crystal lattice, the nature of the crystal size have influenced the company. Thereby, this man is forced to sell the company for the debts what he has made, and he is still working in the company as CEO. You can just, I'm not naming anybody, but this is the real fact, and the company is in Hyderabad. So you can imagine how a small parameter which was ignored by this formulation scientist, like a crystal nature of this particular drug, thinking that all are the same. So particle size, the crystal nature are going to play a very important role. And I give you one more important example of these biological or chemical or physical properties. Majority of you are knowing the cisapride, which is one of the oldest drugs. And if anybody clicks the website of these drugs, any drugs monographs, you can see the statement that it is a practically insoluble drug, especially it is having 0 solubility in 0.1 and HCl. And so far what we have discussed is more the solubility of the drug, more the rate of absorption, because the prerequisite for the drug absorption is the solubility in the respective absorption membrane. Hence, there are at least 1,500 to 2,000 publications on the preparation of solid dispersions of cisapride for improving the solubility. But in recent times, about 8 years back, I have gone through an article where it was reported that cisapride's Tmax, time for maximum concentration, is less than 2 hours, and it is having more than 90% bioavailability. But in that article, they clearly mentioned that the reason for its highest bioavailability with lowest solubility in the acidic medium is not explained. There are no proper mechanisms to explain this peculiar behavior of cisapride. So we come across some such molecules in the dosage form design, and without proper literature support, if you go through that, we are going to land up with the troubles. So when we want to optimize a problem, we have to look into various parameters like poor systemic exposure, poor oral bioavailability, and poor systemic exposure. What is the problem, like distribution clearance or first pass clearance or absorption? So if you just look at this particular table, what is given here, we can just give a question answer like So if one is right, go to the next one. If that is right, you go to the next one. Like that, we go on ending up with the analysis of the drug properties such that we can have a clear-cut idea what are the problems associated for this drug absorption before going into the wet lab for the development of a formulation. We have one more critical approach for the development of a dosage form. All these approaches are called as dry lab techniques, what we call in the modern terminology, because when you get a drug, simply going into the laboratory, doing a trial and error approach, and I'm getting good dissolution, okay, I've prepared a very good formulation is one approach, because there you are not doing any scientific application. Simply by trial and error, we are making a formulation. But in the modern world, especially in the regulatory regime, what we call the trial and error approaches are being prohibited by the majority of the regulators. When you are submitting a formulation for approvals, they are simply asking, what is the rationale of selecting these excipients? What is the rationale of selecting this particular procedure? What is the outcome of selection of this particular procedure, etc.? You need to document very clearly, and then only there is a possibility of getting your product approved by the regulatory authority. So the approach what we now going to discuss is called as LADMER. If you just look at the name, LADMER. So it is nothing but liberation, absorption, then distribution, metabolism, excretion, and finally the response. So the objective of this LADMER is, once a dosage form is formulated, how it is getting liberating the drug. When once the drug is liberated, how it is getting absorbed and how it is getting distributed to the different parts of the body, and by what metabolic pathways the drug is getting degraded, and finally by which metabolic pathway or by which excretory organ the drug is getting eliminated out of the body, and finally after the distribution of the drug, how response is derived, what is called as the therapeutic effect or receptor action or pharmacological action, whatever you call the response, what is required for the patient. So while making this flow chart, we have the horizontal rows called as the biopharmaceutical hurdle, biopharmaceutical class, and approaches to overcome these hurdles. So the first problem what a formulation scientist is going to face is the poor solubility. Because the drug is poorly soluble, which is a biopharmaceutical hurdle, because we consider the physical, chemical, and biological properties, it is a biopharmaceutical hurdle. So it may be as per class 2 or class 4. So we have two approaches, formulation, chemical. So almost all the approaches, they are going to say formulation approach or chemical approach. And when once the drug is liberated, if you are not coming with this particular problem of solubility, the drug gets liberated from the dosage form, and it may be possible that the drug may be degraded before reaching to the site by chemical degradation or enzymatic degradation or due to poor permeability. So chemical and enzymatic degradations are possible for all classes, and we have the approaches for formulation, chemical, or enzyme inhibitors or chemical modifications. And if it is having poor permeability, it may be a class 3 or a class 4 drug. So we have to have sorption enhancers or a chemical modification. And when once it is liberated, it goes for absorption and it goes for distribution. And during this absorption, there is a chance of first pass metabolism, and all classes are prone for its problem, and we may have an alternative route of administration, or by changing the drug into a prodrug, we can overcome this first pass metabolism. But once it is absorbed, and if there is no first pass metabolism, let us imagine there is a distribution of the drug, and it may be directly metabolized after absorption and excreted. That means there is no action of the drug. But after distribution, before exiting the response, it may be metabolized, and there may be a chance of developing an active metabolite which may exert the response. So by distribution, it may give the direct response, or by metabolism also it may give the response through active metabolites. So one can just fit the drug's characteristics one after the other. For this LADMER chart, we can have a conclusive outcome for the approach what a person is supposed to take for formulation of a particular dosage form. And if you look at all these things, chemical modification is one of the criteria that is there for overcoming all these obstacles. But the problem of chemical modification in the new regulatory regime is, even if you change the functional group of a particular drug, a salt formation, again it needs to go through lot of processes of approvals, and it takes a long time for chemical modification to be approved. Hence, the only solution what we have in our control is the formulation approach. That's why always the formulation is going formulation scientists. I'm not making X or Y or Z as specialization. Anybody, a pharmacology person can be a formulation scientist. A pharmacognosy person can be a successful formulation scientist, provided they acquire the knowledge what is required for formulation of a dosage form. So these are the various examples. We have the solid dispersions for poor solubility, eutectic mixtures, micronization, surfactants. These are the various approaches. Chemical modification, of course, it is rarely used, I already mentioned. Chemical degradation, we have to change the pH. That means we always say pH-dependent drug release. By adding some buffers into the dosage form, we try to alter the local pH of the absorbing membrane from acidic to alkaline or alkaline to acidic during the absorption phase of the drug, which may be a cumbersome process and it requires a lot of skill to select a proper buffering agent, etc. And enzymatic degradation, these are the various things that are used. And poor permeability, we have to use the various permeation enhancers. And so what I would like to conclude here in my talk related to this importance of the biopharmaceutical and therapeutic considerations, one need to have the complete pharmacokinetic profile of the drug, biopharmaceutical properties of the drugs. So I'm just taking 1 or 2 minutes extra for making this without a slide. In the present day literature search, majority of the people are banking on search engines like Science Direct, Google, or some other search engines. But if you just look at that particular search engine, you will get thousands of citations with different abstracts. But unfortunately, majority of these are not properly authenticated. So when you want to go for a formulation research, you need to always have an authentic source of information from a referred journal. So abstracts, what we call in scientific terminology, are the best sources. We have the chemical abstracts, international pharmaceutical abstracts, or biological abstracts, or analytical abstracts. These are the scientific abstracts that are published by various societies, forming a consortium of collection of information from various referred journals. So the abstract, what you write in your paper for publication, is going to be abstracted in these particular abstracting services. And once you go into these abstracting services, by giving the keywords what are given in the publication, you can get those abstracts. Of course, nothing is free in the scientific world as per the common nomenclature. So we have to pay something for that. So create a platform. Recently, off the line, Jawaharlal Nehru Technological University, Anantapur Vice Chancellor conducted a video conference on these library services sharing. So he is trying to bring a consortium of the colleges and universities to bargain with these publishers who are making the repeated journals like Marcel Decker publications or Elsevier publications or Bentham publications, etc., etc., such that all the publishers will be on one consortium and the colleges and universities will be in the other consortium, where there will be a sharing of information. In advanced countries like US, UK, and European countries, the consortium of libraries is already established, wherein you are in a place like Minnesota, where you wanted a research publication which is not available in the Minnesota University. So you just go to the library of University of Minnesota and put a requisition saying that this is the publication I want, and leave it to the librarian. Immediately the librarian will go into the search and he finds where the article is available, that means in which library the publication is available. So he puts a request to that particular library with credentials of the researcher who is requiring that particular paper, and within 1 week or earlier, we used to actually, I also received like that from various publishers earlier through post during my early days. But in the modern era, we have an internet. So with a click of a mouse, within 1 to 2 days, the person who requested for that particular publication will be receiving that on a almost majority of the exchanges are priceless, but sometimes it may be a price. But still, it is a very useful approach where we have to generate a strong literature data before going for a formulation approach. That's why if you go to the major companies like Dr. Reddy's or Aurobindo or Hetero or any company you name, which are big players in Indian pharmaceutical market, they have very good subscriptions to all these scientific databases where the people can readily access to all the data, and they prepare a very clear document of the drug file with valid reasons before thinking of a formulation design. And that is the success story of these great industries where they are successful in making a good formulation. For example, you take the latest COVID-19 drug, Remdesivir, majority of the companies have formulated into a injection. They are available in the market. But the trouble, what they have taken in converting this Remdesivir into a vial, like a soluble form, I know what struggle they have faced in converting it. It is a very poorly soluble drug. And converting a poorly soluble drug into a soluble form which is to be injected intramuscularly, which is very, very intravenously, is very, very difficult. I tell you, last but not the least, a small ghastly incident that occurred during 70s. So the people who are terminally ill, especially with diseases like cancer or kidney failures, etc., will be subjected for severe stress and pain. So to overcome this stress and pain, normally in those days, there are now advanced drugs, but in those days, people are administered with digipam. At that time, digipam is given only through intramuscular route or only through oral route. But one company wanted to overcome this difficulty because when it is administered through intravenous route, it may have more immediate action such that the patient will have a soothing relaxation. Though it is not going to induce a direct sleep, it gives some soothing effect to the patient who is suffering with the pain. So they developed an intravenous injection, and it is a blockbuster hit in the market. But people started dying. But nobody recognized what is happening because 90% of the patients are almost terminally ill. So nobody is bothered for their death because they know that they are going to die one day or other. That might be due to the cancer or something with which they are suffering. But one doctor in a particular hospital suspected something wrong based on the patient's conditions. So he took the permission and made an autopsy of these patients who died after ingestion of the digipam injection through intravenous route. And to his surprise, he found the crystals formation in the veins of those patients and found the cause of the death as the blockade of blood supply to the different organs, not because of the disease from which they are suffering. And he brought it to the notice of the administration, and they have gone for more elaborate studies and found that the solubilization mechanism that is used by this particular company for making the diazepam injection, which is a poorly soluble drug, is getting reprecipitated in the blood pH. Thereby, the crystals are getting formed, obstructing the blood vessels. So it is called as digipam syndrome in those days, and later on the injection was banned, etc., etc. But this is the failure of the formulation scientist for not predicting the precipitation of this particular digipam during its intravenous administration, and finally the failure occurred and people died. So sometimes we may be failing like that. So my dear young friends, before going for a formulation development, the biopharmaceutical parameters like physical, chemical, and biological properties of the drugs are to be taken into account, very, very minute details, and then we can proceed to the formulation approach. You will be a great formulation scientist, and I thank one and all for the patient listening of almost 1 hour And I thank once again the Padmavathi School of Pharmacy for giving me this excellent opportunity. Almost this is my last official lecture as principal of AA College of Pharmacy, and I once again thank Dr. Ranganayakalu and the faculty of Padmavathi School of Pharmacy, and Srimati Sulochana, the chairperson of the Padmavathi School of Padmavathi Group of Institutions, and the secretary, I think director of the Padmavathi School of Padmavathi Group of Institution for giving me this excellent opportunity to share my thoughts on this dosage form design and the biopharmaceutical parameters influence on the dosage form.
Thank you very much, sir, for your valuable and very, very useful session, sir. You have enlightened the various mechanisms of drug absorption with practical examples, and you have enlightened the role of each and every factor, that is physical, chemical, biological, pharmaceutical, and dosage form related factors, and their importance in the designing of a dosage form with live examples like the diazepam incident and a small crystallization that affected entire industry, which I have explained just now. Thank you very much, sir, for your valuable session. Sir, with your kind permission, Ramanamurthy sir, shall we move on to the clarification of doubts session, sir?
Yeah, sure, absolutely.
Yes, sir. Sir, a question from Mr. Vasudev Chetty, sir. How can we enhance the absorption of drugs through oral route, thereby increasing the bioavailability of drugs?
See, Mr. Vasudev, the question is very broad. I think I need to take another 2 more classes for giving clarification on this, because there are so many obstacles for enhancing the bioavailability based on the nature of the drug. Is it a poorly soluble or poorly permeable or other characteristics? Based on that only we can give solutions, but not in a broad way, which is very difficult. So I think majority of the answers I tried to give in my presentation, and if anything is there specific, you are most welcome to send me an email, which I will clarify. No worry about that.
Thank you, sir. Another question, sir. Effect of drug interactions influencing in pharmacokinetics, sir.
Yes, we all know the drug interactions may be synergistic or antagonistic. For example, if you take rifampicin, which is my first research work and in MPharm itself and pharmacokinetics, where we observed that para-amino salicylic acid is going to hinder the absorption and elimination of the drug rifampicin, whereas dapsone increases its absorption rate. So we found a synergistic effect with dapsone and the antagonistic effect with para-amino salicylic acid. So drug interaction certainly will influence the pharmacokinetics majorly. There is no doubt on that. We have to take into account individually, but not generally, I mean to say.
Thank you, sir.
Sir, good afternoon, sir.
Yeah.
Sir, this is Kishore, sir, Department of Pharmaceutics.
Yeah, Shanmugam.
Sir, really it is, we are blessed to have a great session, sir.
Pardon?
Sir, we are blessed to have a great session, sir.
Thank you.
Sir, I am having one doubt, sir.
Yeah, please.
So the major problem for bioavailability is the solubility of the drug, no, sir?
Yes.
So for that case only we are preferring solid dispersions to enhance the solubility of the drug.
Yeah.
Thereby we are enhancing the bioavailability.
Fine.
Whereas in the case of nanoparticles, the nano range is the only reason to enhance the bioavailability of the drug, sir.
Pardon, just nano range is?
Nano range is the only reason to increase the bioavailability of the drug, sir. For example,
it is not absolutely, it is not correct. Absolutely, it is not correct. Even for nanoparticles also, solubility plays a very important role. No doubt.
Okay.
Unless the absorption occurs through other mechanisms like the particle phagocytosis type of engulfing the nanoparticles by the cell endocytes and then transportation of these nanoparticles to the other side of the membrane. For all other nanoparticles, solubility is also very important. But the advantage of nanoparticles is the highest surface area, whereby the solubility will be increased to a great extent compared to the normal drugs. That is the advantage.
okay, sir. And actually, because of that nano range, it directly gets absorbed into systemic circulation from the G18, no, sir?
It all depends on the part nano size. That is very, very important. Not all the nanoparticles. For example, if the nano size is less than 200 nanometers, there is every chance of these nanocytes being absorbed through the endocytosis. But others cannot be absorbed if their nano size is more than 200. Normal rule. That's it.
Okay, sir. Thank you, sir.
Sir, good afternoon, sir.
Good afternoon.
Sir, I have a general question, sir, with regard relates to this corona vaccine.
Yeah.
So people in and around are very eagerly waiting for this vaccine, sir.
Yes.
We know the product to reach into the public, it will take minimum of 15 years to identify a drug and to make it as a dosage form.
Right.
So what kind of trials are doing to accelerate this product to bring it out?
See, whatever type of trials they are going to do, according
Sir.
to my phase 1, phase 2, phase 3 trials knowledge,
Sir.
I feel by next April, May, only we may have a vaccine for the initial application on the human volunteers. That
Thank you,
also because earlier in any phase 1 or phase 2 or phase 3 clinical trials, after completion of a phase 2 trial, they used to take minimum 1 to 2 years for analysis of the data. Because they have to go through all various parameters to see that there is no side effect of anything at any point of time. Because when I have seen the phase 2 trial records in a company called as Malincraft in USA during my visit to USA, even a person who complained a slight dizziness, that case was reviewed for 10 days, only that particular volunteer case, by a team of about 15 experts comprising of various fields, medicine, pharmacy, biochemistry, etc., etc. And they used to have a meeting right from 9:30 or 10 o'clock in the morning till 5 p.m. or 6 p.m. without a break to analyze what could be. They used to go for all vital biochemical tests and they used to analyze what is the reason. And then only they have cleared that, okay, it is a small complaint without any valid reason, and hence they cleared. Like that, they used to do a lot of exercise for clearing the phase 2 trials before submitting for the regulatory approval. Because once it goes into the regulatory hand by the company, they have nothing to do with that, and all the decisions what are taken by the regulator is going to be the final. So, majority of the companies are so careful in giving the data, clinical data, what is generated by the company to the regulator, and they do a lot of exercises for not less than 3 to 4 months or even 5 months. So that is the time normally. That is a delay with which we are getting the delay of the drugs into the market. But with the COVID pandemic, both the regulators as well as the researchers are just pushing certain things without giving much light into the concepts what they are expected to do. And that may positive or that may be negative also in future. Like for thalidomide, you take the thalidomide in 7 days, where the drug was very hastily introduced in the market without proper understanding of the effects and the drug density, and it caused a lot of havoc in the market. So in a similar way, if it is positive, everyone will be happy. If it is negative, again, everyone will be happy. There is
Yes, thank you, sir.
Thank you very much, sir. One last question, sir.
Yeah.
Sir, can you please brief out how can we apply these formulation techniques to
Yes.
herbal drugs
I given
to overcome the absorption factor?
earlier talk on herbal drug formulation also.
Sir.
Frankly, compared to the chemicals which we call as drugs, herbal drugs are much more problematic because we cannot identify a single conscience responsible for the therapeutic effect. Majority of them are extracts containing different chemical compositions, and these chemical compositions are going to alter from the nature of the extraction, site of the sample that is collected, and other parameters. So if you get an extract for formulation development now, and if you get the second batch of the extract, again that may affect your initial formulation trial. So we need to have more validations compared to the pure chemical drugs in the herbal drug dosage form. That is my personal experience even.
Yes, sir. Yes, sir. Thank you, sir.
Yeah.
Sir,
yeah, I think Gopinath, the example of cisapride is the best for this answer. There are certain mechanisms by which the drugs are absorbed, and there are no explanations so far. They have not given the reason, valid reason. Though there is no solubility, the drugs are having very good bioavailability. Still.
Yes, sir. Thank you very much, sir, for clarifying doubts. Now I request convener of today's session, Dr. D. Ranganayaklu, sir, to address the session. Sir, please.
It's my pleasure and privilege to give this back. Really, I am very thankful to my professor to avail this kind of opportunity where sir is very much announced this is a last official webinar program. Thank you very much, sir, for considering this request and gracing our institute as well as updating our students in the regard of subject wise. And bring back my position. I'm able to recall last 23 years back when I'm there in the campus doing my pharma. Really, I felt very happy. The same, what you can call thoughts has been generated. How basics has to be updated to more research and other formulation side. Really fantastic, because though it is a basic one regarding the pharmacokinetic parameters pertaining for solubility, lipid, metabolic solubility, definitely have the lot of influence has been explained. Because these have been adopted for this absorption. Absorption go to place the absorption. When the absorption is over by biochemical, biotransformation, so one has to be assess the ability of the bioavailability, because that is only the purpose. Without any bioavailability, maximum mark, there is no kind of any curative and therapeutic value. Certainly, definitely justice that all the topics of biokinetics, one has to relay in part of any allied lines like cognition, ecology, biopharmaceutical kinetics parameters are very varied. Definitely, our students has been updated and the scholars has been ignited towards the research activity. Thank you very much, sir, for considering our request and sparing your time, busy schedule, and keeping aside and raising our institute. On behalf of institute, once again, I convey my thanks on behalf of all the pharma buds and young teachers and other senior teachers for kind of availing this kind of opportunity at the live webinar program. At the same time, on behalf of the institute, on behalf of the student, on behalf of my faculty, and my personal side, I pray the mighty, that is Lord Sri Venkateswara Swami, has to provide much more peace and harmonial function of superannuation. Definitely that your things are going to be reflect for that, no doubt in that. But even then, my prayer to the Almighty to provide much more the peaceful and healthy, wealthy, prosperity in the coming 40 years. We definitely your support and timely advice to be required for the institute, though we may be retired also. This is my little expectation from your end, sir. Thank you very much. At the last end, now it's a token of respect. We have a formality. Please kindly accept the token of appreciation, sir. Thank you very much.
Thank you very much, Ranganayaklu. We are with you always.
Thank you, sir. I pray
very much. Yes, sir. Thank you very much.
Thank you.
Next, I request coordinator of today's program and head of Pharmaceutics department, Dr. V. Shanmugam, sir, to please deliver the vote of thanks for today's session. Sir, please.
Thank you. Thank you, Srinath. Respected today's speaker, dignitaries, teachers, and participants. Good afternoon, one and all. On behalf of Sri Padmavathi School of Pharmacy, I, Dr. V. Shanmugam, take this opportunity to propose vote of thanks to those who contributed to this webinar on biopharmaceutical and therapeutic considerations in dosage form design, organized by the Department of Pharmaceutics. At the outset, I thank our honorable chief guest and speaker, Professor K.V.Ramanamurthy Garu, Professor and Principal, Andhra University, for accepting our request. We are really enlightened with your knowledge and gracious presence, sir. We are thankful to our Honorable Chief Patron, Srimati P. Sulochana Madam Garu, Chairperson of Sri Padmavathi Educational Institutions, and Patron Sri P. Pranith Garu, Director, SPI, for their motivation and support. I would like to thank our convener, Dr. D. Ranganayakul sir, and organizing secretary, Dr. C. Sridhar sir, HOD, Departmental of Pharmaceutical Chemistry, for your enthusiastic support in this webinar. A special thanks to the organizing committee, teaching and non-teaching staff for their unflinching support and coordination. Our heartfelt thanks to our students, delegates. Around 400 registrations from different parts of the country and world today have actively participated in this webinar. Last but not the least, I extend my sincere thanks to press and social media for spreading and projecting our session to the student places. With this warm words, a kind message, we move to the end of today's seminar. Thank you.
Thank you.
Thank you very much.
Thank you very much.
Thank you, sir. Thank you. Good day, everybody.
Thank you, sir.
Thank you very much, Dr. Shanmugam sir. Dear participants, hope you all enjoyed the session with much more useful information regarding the theme of today's session. We have already forwarded the feedback forms to your respective mails and also posted in the WhatsApp group. So we kindly request you all to fill the feedback form so that you can avail the e-certificate and automatically will be generated to your mail. Thank you very much, all the participants. And please be patient. If you don't receive the certificate today, definitely you'll get it tomorrow, as there is a limit for generation of e-certificates. Definitely you'll get in a week time. Thank you very much, dear all participants across the world, for your active participation and making this program a grand success. Once again, thank you very much, Ramanamurthy sir, for accepting our invitation and being with us today. Thank you very much, sir. Dear sir, now we all join for national anthem, sir.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you.
Thank you, Ranganayakul.
So,
Bye.
yeah, we are very enthusiastic to attend the 29th program, sir. Just pass out the password.
Sure, I'll share it. No worries. Absolutely. Right.
Yes, sir.
Thank you. Thank
Sir,
you.
with your thank you. Shall we end the session, sir?
Yeah, sure.
Thank you very much.